Thank God for the creation of viruses. A few more reasons why, and I'll talk more about the coronavirus in a minute. They serve as vectors, and that would be for transferring a gene from a lab or where else, from another plant, into another plant or another animal. So they're vectors. And Lambda virus is used because it's huge, and they can pull out lots of genes, and the virus still does its job, and they can load lots of other genes. So they can be carried into wherever they want to carry them. And by the way, that's what viruses do. They get inside the cell and they drop off their genes and put them in your DNA. They splice your genes at the right place. They can't just splice them anywhere. They splice them at the right place. They know where to splice them. They splice it, they put in the genes, and they fix the ends. So they combine the two together so your body thinks it's just another gene. You have to do all this. And you have to have the coding sequences, you have to have the introduction, the start codons, and the stop codons. So all this has to be put in. So it's not simple, it's very complex system. And what else can viruses be used for? Well, one, it has the potential to cure cancer. If they can only identify the ID tags on cancer cells specifically, so the viruses only attack the cancer cells and no other cells. The goal is you could have stage 4 cancer, you're dying, your liver's gone, it's in the bones, it's in the blood, it's everywhere. These viruses then will hunt out every single cancer cell, destroy every single cancer cell, and you will be free to go home. You have no cancer whatsoever. They're all destroyed. So that is the goal. The problem is you've got to make sure they destroy the cancer cells and not the other cells. So we have to get systems that affect only the cancer cells and other cells. Because cancer cells, you know, are your own cells. But they are changed as a result of the cancer, and that's what we have to identify. And we can also use them to fight other viral infections, pathogenic infections. And we use viruses, of course, in many studies to determine molecular mechanisms. In fact, biochemistry would be out of business if it wasn't for viruses. Viruses are critically important in doing research. When I worked at the medical school in the lab, we used viruses with just about everything we did. Very important. The coronavirus, that's what people are wanting to hear about because that's causing a lot of deaths, as we know. And the coronavirus, they have spike proteins, these things coming out, these identification tags, and they're multifunctional molecular machines that mediates the coronavirus entering the host cells. To explain that, many viruses, to get in, they have to have 2 keys. They have 2 identification tags. And they can't get in unless they have both. AIDS is the one I know best. AIDS has a CD4, I believe, and a CD44. So to get into your cell, the cell must have the 2 entry points. It must have the CD4 and the CD44. It cannot get in unless the virus has both of those tags, both of those licenses. If one fits, can't get in. If the other fits, can't get in. It must have both. So it's to some degree like the key to your car. If I had a key, chances are it would not fit in your car. It's not designed to fit. That's designed not to fit, actually. And the same thing with viruses. Viruses are designed to fit inside of certain cells. When it has the right surface, the subunits, and the right key, it gets in, does its job. So you can see that the design is perfect, and of course we know it's not perfect today because of the fall. So this is explained all very well by the fall.